can say that immune system is body's defense system, which helps us to combat infections, allergies, and any pathogenic organism. Immune system is divided into two portions: innate immunity, that is non-specific or inborn immunity, and adaptive immunity. Adaptive immunity is a specific immunity and it is acquired immunity. Further, adaptive immunity has been divided into humoral, that is antibody dependent, and cell mediated immunity. In cell mediated immunity, we just focus on T cells and B cells. Innate immunity, that is inborn immunity, is, first of all, we talk about the anatomical barriers, that is skin, mucous membrane, air vacs, tears and eye. So all those barriers make sure that no foreign pathogen is able to enter inside the body. But still, if because of any cut or the pathogen is able to enter inside, so the main functions of the immune system is it should be able to identify self and non-self. It should have non-reactivity to self particles, and this property is called tolerance. And non-self refers to particles that are not made by our body and are recognized as potentially harmful. They are also called foreign bodies. So the foreign pathogen, which is foreign pathogen, bacteria, virus, anything which is trying to enter inside the body and is not non-self, is called antigen. Antigen is anything that causes an immune response and can be entire pathogen like bacteria, virus, fungi, and parasite, or it can be a particular part of it. There are few more factors of innate immune system, including cytokines. For example, interferons are one of the examples of cytokines. We have multiple other examples like interleukins, growth factors. These molecules are used for cell signaling or cell-cell communication. They are used to communicate with neighboring or distant cells about initiating an immune response. Let's talk about cells of the innate immune system. There are a lot of cells which are involved in innate immunity. Innate immunity basically makes sure that till the time adaptive immunity is activated and the B cells, T cells activate and they produce antibodies, meanwhile, pathogens are not able to do much harm to the body, and that's why cells of the innate immune system come into action. First, we have phagocytes. Phagocytes are very big cells. They are much bigger than the normal cells, and the way by which they act is called phagocytosis. That means they engulf the foreign pathogen. That's why we also call them eating cell. If they engulf a large pathogen, the process is called phagocytosis, and we call it cell eating. And if we talk about very small cells being ingested, then it's called pinocytosis, and in other terms, we also call it cell drinking. Phagocytes circulate throughout the body looking for potential threats like bacteria, viruses, and they engulf and destroy them. For example, macrophages. Second, we have mast cells. They're important for wound healing and defense against pathogens. Dendritic cells are antigen-presenting cells that are located in tissues. They are just like, they are like detectives. They just collect evidences. They will collect pieces of antigen and they'll present it to the B cells and T cells, the factors of the adaptive immunity. Eosinophils are granulocytes. They have granules on them that secrete a range of highly toxic proteins and free radicals that kill bacteria and parasites. Now the adaptive immunity. When an antigen enters the body and lymphocytes create antibodies to fight against them, this is called adaptive immunity. We have divided this into humoral, that is antibody formation, and cell-mediated immunity. In cell-mediated immunity, we basically talk about B cells and T cells. So a link between innate immune system and adaptive immune system is dendritic cells, because dendritic cells come and present the antigens, that is fragments of antigen, to the T helper cells. Then these T helper cells do a crosstalk with B cells so that plasma cells, that is antibodies which are short-lived and going to act instantly, and memory cells are formed. Two types of cells of immune system: B cells, which are secreted in the bone marrow, and T cells, which are formed in the, they're also secreted in bone marrow, but then they are transferred to the thymus. Talking about the organs of immune system, there are two types of organs: primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs just like bone marrow and thymus, secondary lymphoid organ like spleen, and then we have many other, gall, mal. B cells produce antibodies, whereas T cells promote the destruction of microbes residing in phagocytes or killing of infected cells to eliminate reservoirs of infection. There are 5 types of antibodies which are found. IgM is usually called the primary response antibody. IgG is the one which is formed in our body as a memory antibody. IgD, the function is not much known. IgA is found in a lot of secretions like breast milk. And IgE is antibody against infections like allergens. T cells do not produce antibody molecules, but they have antigen receptors that are structurally related to antibodies. These structures help recognize antigens only in the form of peptides displayed on the surface of antigen-presenting cells. Antigen-presenting cells basically are a group of cells, a fragment of the antigen, and present it to the T helper cells so that these T helper cells go and talk to the B cell for antibody formation. Antigen presentation has been divided into few steps: pathogen recognition, phagocytosis of the pathogen, processing of the antigen so that a small fragment of antigen can be collected, and then presentation of that small fragment to naive T cells. T cells must be presented with antigens in order to perform immune system functions. The T cell receptors is restricted to recognizing antigenic peptides only when bound to appropriate molecules of the MHC complexes on antigen presenting cells. T cell receptors are going to recognize only antigenic peptides, those which are foreign, and they are bound to molecules of MHC complexes. So if antigenic peptides are bound to MHC complexes, only then they will be recognized by T cell receptor. MHC can be of two types, MHC1 and MHC2. MHC molecules which present antigen to CD8 plus T cytotoxic T cells. Basically, T cells are of two types, T helper cells, and then there is other type called cytotoxic T cells. How to distinguish between these two cells? So we have put a marker over there. CD4 is a marker for T helper cells. So when we say CD4 T helper cell, that means we are talking about T helper cells. And when we say CD8 plus cytotoxic T cell, that means we are talking about cytotoxic T cells, killer T cells. Class 1 MHC is expressed by almost all host cells. Cytotoxic T cells are specialized for inducing the death of other cells. Now, recognition of antigenic peptides through class 1 MHC by cytotoxic T cells leads to killing of the target cell. Whereas on the other hand, T helper cells present their antigen to B cell, which will activate their proliferation response. Typical structure of an antibody, also called immunoglobulin. These are quaternary structure. They have two heavy chains and two light chains. This is one heavy chain, another heavy chain, and this is a light chain, another light chain. They're held together by disulfide bonds. This region is called epitope, which is meant for antigen binding. Now, there are two regions on the antibody, variable region and constant region. So we have variable heavy chain and constant heavy chain. Similarly, we have a variable heavy chain and a constant heavy chain. Variable light chain, constant light chain, variable light chain, constant light chain. Antigen is any molecule that induces an immune response, such as toxin or molecular components of a pathogen.